Okay, welcome to the next installment of the EOC Biology Review at Spanish River. Today's concept is genetics. I'm Miss Jo. Let's get started. So first we have to recap on some vocabulary. Primary terms here, genotype versus phenotype. Of course, the genotype is the genetic combination of a trait. Remember that all individuals contain 2 alleles for each trait, and genotypes are represented by alphabetical letters. For phenotypes, they are adjectives. They are the physical expression from that genotype. In terms of traits and alleles, traits are going to be the characteristics that could actually be passed from parent to offspring. An example of that in humans would be eye color. And associated with each trait, we have 2 possible forms of an allele usually. So an example here in terms of eye color, again, would be blue eyes in humans. Remember, again, most traits have 2 alleles for each trait. In terms of those alleles, we have the law of dominance, which states that one trait is usually stronger than the other, and that's going to be represented by the capital letter. And of course, then we have a recessive, which is the weaker, and it's represented by a lowercase letter. When we have those combinations of alleles, so we have 2 letters going on, we have 2 different options. One can be homozygous, which is a gene combination involving either 2 dominant or 2 recessive genes. In either case, they will be the same allele. And then we have heterozygous. Hetero means different, so they're going to have 2 different alleles, one being dominant and one being recessive. And finally, we just have to recap really quickly the difference between males and females, of course. Males are going to contain an X and a Y chromosome, which means that sons are going to inherit their X from their mom and their Y from their dad. And then we have females, which contain 2 X chromosomes, one from the mom and one from the dad. If you see here our test cross, we always have the bottom 2 squares expressing our sons or possible males, and the top 2 expressing females or possible daughter offspring. Also, we have to really quickly review what a Punnett square is, and that's going to be a visual representation of a test cross. So basically, we're going to be predicting what our offspring's genotypes and phenotypes will be. There are 2 primary types of crosses. The first one is called a monohybrid cross. Mono means 1, so we're crossing 1 trait. And then we have a dihybrid cross, which crosses 2 traits. Our first example here is a monohybrid cross. We are crossing 2 heterozygous plants for pea color, and you'll see that both of our parents have the yellow color, and because they're heterozygous, we know that that means that our capital letter here, Y, stands for yellow, and our lowercase would be the recessive trait of green. Keep in mind that there is 1 allele in each box for test cross. Once we cross these, we will see that the letters intersect to create our possible offspring in our boxes, and our genotype ratio is 1 to 2 to 1, meaning there is 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. That genotype then expresses in the phenotype. And if you look at our phenotypic ratio, you will see that it is not equal to our genotypic ratio, and that is primarily because we have the rule of dominance. So if you have a heterozygote, which means you have at least 1 dominant trait, you of course express that dominant trait. So here we have 3 offspring, the yellow color, and only 1 homozygous recessive expressing that green trait. So our ratio is 3 to 1, 3 yellow and 1 green. Second type of cross is a dihybrid cross. We're going to be crossing 2 different traits. So here we are going to go back to our pea plants. They are heterozygous for pea color, yellow and green, which we discussed before. And now we're going to add our second trait, and that's going to be shape, either round or wrinkled. And again, round is capital, so you know that's the dominant trait, and wrinkled must be the recessive trait then. So if you see our genotypes, we have capital R, lowercase r, capital Y, lowercase y, because again, we're heterozygous for both of those traits. These are our possible gametes we can form. And this is color-coded for a reason, because we are going to use this FOIL method, and we're going to show how those gametes are formed. The first step is you take the first of your alleles and combine them. So we have our red RY there. Then you go outer, capital R, lowercase y, your inner, lowercase r, uppercase y, and then our two recessive traits are and y, both lowercase. So that's how we form these possible gametes. And basically from here on out, a dihybrid works exactly the same way as a monohybrid. You just have to keep in mind that we have one allele for each trait on the side of each square. So you can never have two of the same letter over or to the side of the same box. So please keep that in mind. So if we have our possible gametes, and again, we're crossing two heterozygotes, so they're going to be the same on both sides there. Once we set up our crosses, again, it works exactly the same. So I know that I have homozygous dominant for round, homozygous dominant for yellow here because of the intersection between those two gametes. Here we have both capital R's, but you see our recessive traits have been added there, and so on. So in a sense, if you take our genotypic information and transfer it into our phenotypic information, then we are going to get a 9:3 to 3:1 ratio. Basically because we have 9 that are round and yellow, we are going to have 3 that are round and green, 3 that are wrinkled and yellow, and only 1 here that's going to express the recessive traits of both shape and color of peas. Okay, before we get started on some patterns of inheritance, we have to discuss 2 laws of Mendel. And of course, Mendel is the Austrian monk that is coined the father of genetics. The first law we have to look at is called the law of segregation. And that law basically states that alleles are separated from each other during gamete formation, and they're recombined at fertilization. And this little diagram down here basically exemplifies that. We have a diploid adult who, of course, has reached puberty because through the process of meiosis, they produce their haploid gametes. And we know that when you put 1N plus 1N, you come back full circle to 2N. And, of course, fertilization is the step that makes that happen. Essentially what's happening here is that you have the formation of gametes, and gametes only have 1 allele, but we know that full functioning organisms have 2 alleles for each trait, and those 2 are recombined based on the process of fertilization. Keep in mind that's, of course, through sexually reproducing organisms. And we do have 2 alleles, and we're going to get 1 from each of our parents. This is essentially the same thing in a picture. So we have a homozygous dominant purple flower that was crossed with a homozygous recessive white flower. And so you go from 2 alleles to 1, recombined here at fertilization. And those 2 traits now create a diploid organism, but this purple flower here, even though it's heterozygous, still produces just 2 gametes, or 2 gamete possibilities, and we will still have to split up each allele. So, long story short, law of segregation. Diploid, you have 2 alleles. Haploid gametes have 1. They are recombined at fertilization. The next law we're going to look at is independent assortment. And basically all that states is that our allele pairs are going to separate independently during gamete formation. Again, that process known as meiosis. So essentially traits are passed independently of another. Long story short, you don't pass on what your mother gave you in whole. You pass on one portion of what you have received. So just because your mother or father passes you on a combination of alleles, you still pass on one of those alleles for the test cross. And this is a key in increasing genetic diversity among organisms. Again, you must sexually reproduce in order to receive this benefit. So if you look at this process through a diagram, again, we're going back to our original pea plants here. So we have homozygous for shape and color and heterozygous for shape and color. They can still only create the same amount of gametes as everybody else, no matter how many traits they have. So here we are going to become or separate out into one gamete. And this is our possible offspring here. If you cross two homozygous, one recessive, one dominant, you're pretty much going to get offspring that are heterozygous for each thing. And this heterozygous pea here, this yellow round pea, is not going to pass on the exact traits that his parents passed to him in terms of the test cross. That is never happening. You still have to use that FOIL method in order to see which gametes that individual can pass on to his future offspring. So again, if you go first, outer, inner, last, they are the order here. So just because you're given a grouping from your previous generations, that grouping is not passed on. They are independently assorted. Okay, so let's look at some patterns of inheritance. The first one we're going to look at is the law of dominance, which basically just states if you have at least one dominant allele, then you're going to express the dominant trait. This is pretty much a review since we've already done this cross essentially. So if we have two heterozygous plants for plant height, that means that we have each plant having the two different alleles. If we do our test cross here, the genotypic ratio of our offspring, if you see here, we have one homozygous dominant, we have two heterozygotes, and we also have one homozygous recessive. The percent of offspring that are tall. Remember that if you have one dominant trait, you express that dominant trait, which means that we have 3 out of 4 individuals that are tall, which would leave us with 75%. This principle, which genotype can never produce offspring that express the recessive phenotype? Think about it. If you have to have at least one dominant trait to be dominantly expressed, the ones that can never express recessive are going to be homozygous dominant. No matter what they're crossed with, even the homozygous recessive, there will at least be one capital letter in each box. So homozygous dominance can never pass on enough alleles for their offspring to express the recessive trait. The next example here is codominance. And basically that's going to be when the phenotype is produced by both alleles are clearly expressed. Common examples, roan fur in horses, permanent feathers in chicken, and human blood types. If we look at a human blood type question, we will see that these are our human blood types here, and they are definitely going to have multiple alleles since we have 3 options here, which produce 4 blood types. And if you look at our blood typing, here's a trick kind of practice question for you. If you have a boy that has blood type O, and remember that is going to be our lower boxes, females on the upper boxes. So if our son here has blood type O, then I know at least one box looks like that. If we have a sister here that has blood type AB, and she's female, she goes up here, I know she has to look like this. It is a codominant trait. So what are the genotypes and phenotypes of her parents then, or their parents? We know that they have to at least both carry the recessive trait, which is part of the type O. We also know that they have to, no matter the gender, carry both the A and the B. So essentially these are the genotypes of your parents. And the phenotypes, of course, this phenotype would produce type A blood, and this phenotype produces type B blood. All right, sex-linked traits are termed that and also commonly called X-linked traits because they do carry most traits on that X chromosome. And so basically females can rarely express that trait unless they are homozygous recessive. Think about it. They get one X from mom, one X from dad. So dad essentially has to have whatever the trait is, and they're going to have a mother that either carries it on the left-hand side of your screen or is affected on the right-hand side of the screen, because the only way for a female to get this trait is to have 25% chance there, 50% chance here in terms of the females. This one's going to just be a carrier. So if we move on, we will see that we have common examples: eye color in fruit flies, human hemophilia, and human color blindness. So if we crossed a red-eyed male with a white-eyed female, of course this is for Drosophila, we would have to have a male with red eyes, and again, it's only carried on the X, right? Our Y chromosome does not carry that trait, so our X will carry it, our Y will not. And we have white-eyed female, and again, if a female shows or expresses that recessive trait, she must be homozygous recessive. So if we perform our cross here, we will see that the only possibility of eye color for females, if you switch back to that screen a little bit, is going to be red eyes, because the male carries the red eye, which means she's going to get at least one of those X's in each box. So even though the other one's recessive, dominant rule still applies. What is the only possibility of eye color for males? Again, the Y carries nothing, and our X both have a white allele. So the only eye color for sons essentially are going to be that white eye. If you switch over and look at our pedigree here, while I clean this up a little bit, this shows you basically inheritance patterns. It doesn't really matter what the recessive trait is. In either case, if you look at this pedigree, you will see that we have an affected male here and a not affected female. However, we have an affected male here. So right away we know it's a sex-linked trait because there are more men in this picture that are affected. So what must be true for the females in generation 2? Here. We know it must be true because they have a father who's affected. So we know that that father is affected. The mother, we don't really know much about that mom, but honestly it doesn't really matter, because if we have an affected father, then we know no matter what the other X is, and actually we know the other X because we have no females here that are affected, so we know that the mother could not have been affected herself. Maybe she's a carrier. Doesn't really matter. In either case, we ended up being that square right there. So we know for a fact that all females in this pedigree must be carriers. So, second question, did the father of male 2 in generation 3 have any effect of the expression of this trait? The answer is always no. Nothing's carried on the Y chromosome in terms of a sex-linked trait, so it doesn't matter what the father carries, he has no difference or no effect on the expression of his offspring. All right, two final points here. We have multiple alleles, and those are going to be genes that have more than two alleles, because again, most have two for each trait. Common examples, human blood type again, and fur color in rabbits. Remember that individuals still can only have two alleles per trait, but within a population's gene pool, we can still have more than just those two possible alleles. In humans, it's commonly represented by the capital letter I, and again, you can refer back to human blood type examples to show you that. If you look here at rabbit fur, you see we have these four possible alleles for the same trait, which is rabbit fur color. And finally, we have polygenic traits. Common examples of that you might see on the EOC are reddish-brown eye color of Drosophila, again, fruit flies, human skin color, and human eye color. And polygenic traits are essentially just traits that are controlled by two or more genes. These often show a wide variety of phenotypes, since there are multiple different characteristics or ranges that can be produced in one organism. So you see here the progression of human eye color from blue to brown, and the myriad of skin colors that are possible on this planet as well.